Well, welcome everybody. My name's Darren Crane. I'm the Director of the Australian Tropical Herbarium, and Katharina Nargar is one of our staff, and she'll be giving a wonderful talk today. But first, I'd like to acknowledge the traditional owners of the country on which we're meeting today. And here at James Cook University, we acknowledge the Australian Aboriginal and Torres Strait Islander peoples of this nation, and particularly of the lands on which we're meeting, the Jabberkai, the Yurrugunji, and the Gimwi Yidinji people. We pay our respects to the ancestors and the elders of those Aboriginal nations, past, present and future. So let me just introduce Katharina. I've had the very great pleasure of working alongside Katharina since 2010. And so Katharina's research is mostly around molecular systematics and evolutionary biology, with a particular focus on tropical plant diversity. And when we first met, it was when we were both working on bromeliads, of which we have none in Australia, but nonetheless, that was our first connection before we came together here at the herbarium many years later. So Katharina's research employs high-throughput DNA sequencing approaches to understand the diversification of species-rich plant groups in time and space, and the factors that have shaped that diversity, the evolutionary factors. So a lot of her work is around conservation genomics, particularly around assessing species boundaries and what that means for determining units for conservation and genomic diversity, again with a focus on rare and threatened species, particularly orchids. So she's been with us at the Australian Tropical Herbarium as a research scientist since 2010, but her actual employer is the CSIRO, and she's attached as one of the CSIRO's contributions to the Australian joint venture. So I'll hand over to Katharina.
Thank you, Darren. Yeah, thank you everybody for coming to the seminar this afternoon. So Lawrence suggested that I present again. I've given a talk maybe 3 years ago, just to make sure that our students know what's happening at the herbarium. So I'm going to talk about our collection genomics work at the herbarium, focusing on orchids, their evolution and conservation, and especially of the Australian orchid diversity. I want to start with the latest release of the Q State of the World's Plant and Fungi report that was released this week, which found that up to 45% of flowering plants are under threat of extinction. And orchids are a group that are particularly affected of this. Biological diversity is under enormous pressure, as we all know, from climate change and other threatening factors such as habitat loss. However, the challenge is that we still lack fundamental biodiversity data. And one of those challenges is around our insufficient knowledge about how many species of flowering plants there are. And the estimate is about 100,000 remain to be named. At the current rate of about 2,500 new descriptions each year, that would take us 40 years. And clearly, we have to find ways to accelerate the research that collects fundamental biodiversity data. Biodiversity data is fundamental for conservation, and this is a lovely chart that is also included in the Q report, which starts with the fundamental data of plant names and locations. And these are then fed into curated biodiversity databases that add data like trait data, genetic data, and ecological data, for example, and then inform evolutionary studies and feed into extinction risk assessments, diversity estimates, distribution maps, and then help to design protected areas. So, contrary to the more romantic notion of a botanist walking through the field and discovering a new species, it is the true work of the botanist usually to work in botanical collections focusing on a particular plant group, and then by comparison, find new species. So herbaria and botanical collections are major frontiers of species discovery, but they are also a really critical research infrastructure. They are rich repositories of biological diversity with over 350 million plant collections worldwide. And they provide a temporarily deep and spatially broad time series of samples, which allows us to look back into previous genetic diversity, for example, of past populations or even extinct species. They also represent an investment that has been built over centuries by many generations of scientists and botanists and hobbyists who collected plants throughout the world, which now gives us the opportunity to have all this aggregated information at our fingertips to be ready to be studied in more detail. Our collections are very precious, and when we work with them, we want to be as least invasive as possible, and that is also applicable to DNA studies. So we can only afford to take the smallest amount of samples from those precious collections, and challenges around working with those specimens for DNA research is that DNA is degraded and contains contaminations from other organisms that have started growing or have grown in those specimens, like fungi. However, with new DNA sequencing technologies, those DNA degradations and contaminations are now manageable, and even for DNA degradation, almost an advantage, because the new technologies require small fragments of DNA to be sequenced high throughput. The contamination is dealt with bioinformatically quite elegantly today. So our botanical collections in Australia host around 4.5 million collections of Australian flowering plants. And high throughput sequencing has really been a game changer in the mobilization of those collections and making those genetic collections also digitally available for the whole world to use. So a technique that I want to introduce that has been really revolutionary for DNA research is target capture sequencing, or also target enrichment is another term for this. And it is an ingenious strategy to only sequence those areas of interest. So the genome is made up by lots of repetitive DNA that is barely informative, and we are really only interested in the regions that are coding, which is the smallest part actually of the genome. And so sequencing the whole genome is very wasteful. And so target sequence capture uses hybrid probes that are of the target sequence that we are interested in studying, and attaches, hybridizes to the DNA strands in the DNA mix. And then by putting in magnetic beads that can attach to those baits, we can magnetically enrich the sample and tip out all the other liquid that doesn't contain our target DNA, and then sequence that. The method is quite elegant, as it allows us to capture any part of the DNA that we are interested in, and in large quantities, so many hundreds and thousands of markers. It is really interesting because it allows for a design that can capture different depths of evolutionary distance, so within a population, but also within genera or families. And it is able to address some tricky features of plants like polyploidy or hybridization. So there are several large-scale important collection genomic projects underway nationally and internationally, and I want to highlight two of those. The Genomics for Australian Plants initiative, which aims to build the Australian angiosperm tree of life from hundreds of nuclear markers. And in its current stage, that is just about to complete, the target was to build a genus-level phylogeny for all Australian, for 90% of Australian genera. The second, and that is an international initiative, is the Plant and Fungal Tree of Life initiative, which builds an evolutionary tree for all flowering plant genera. And of course, that overlaps with the Australian initiative. And so the two initiatives have partnered. In the last data release, the GAP consortium contributed over 750 Australian species to this effort. The Australian Tropical Herbarium is very invested in the GAP consortium, and Darren is leading the phylogenomics component of that project. So now I would like to turn to our research on Australian orchids and give a bit of an overview introduction to Australian orchids. So first of all, orchids are the second largest plant group on Earth with about 30,000 species and hugely diverse. In Australia, we have around 1,600 species, of which more than 90% occur nowhere else. And Australian orchids also comprise a large proportion of Australia's threatened flora, with about 15%. So the orchid family tree globally is quite sculpted. We have 5 main evolutionary lineages, and this is an artist's impression, with a small branch of lily-looking orchids at the base. Apostasia is a genus after it is named, and we have Apostasia growing just here in our rainforest. You don't have to walk more than 10 minutes to see them there. Then the vanilloids with the vanilla, also not very speciose, and we also have some native species of that group in Australia. Slipper orchids, named after their peculiar flowers that look like a lady slipper, don't occur in Australia. They are more an Asian temperate element. And then the second largest branch are the orchidoides, which are terrestrials and have around 3,100 species. And then the large branch of the epidendroids. Epidendroid means growing on trees, and this is a mainly epiphytic clade and usually diverse and mainly concentrated in the tropics. Recent DNA studies estimated the age of the stem of the orchid tree around 120 million years ago, so very old. Navigating a group that is so hugely diverse is really difficult, and orchid classification helps us to find our way much more quicker by the different hierarchies, starting here from the subfamilies and then into the different lower categories, taxonomic groups. This is a graphical representation of the same tree that I just showed you, and it is important to understand how to read it. So the first branch here is the first diverging lineage, and then the next one and the next one. And the ones that are grouped at one node are most closely related to each other than ones that are more distantly positioned. So the Australian orchid family tree looks different from the global orchid family tree, and it's very distinctly so. The epidendroid branch that contains the epiphytic orchids is usually reduced, mainly due to a lack of suitable habitat here with our much drier climate and less rainforest than other parts of the world. In Australia, the Australian wet tropics is the most species-rich area for Australian epidendroid orchids. Australia has a large proportion of the terrestrial orchidoids, and in fact harbors about a third of the world's diversity of this group. And they're characterized by underground tubers that facilitates those orchids to stay underground in times of inhospitable seasons like the Australian summer. Australian orchids are mainly distributed along the rainfall gradient, and that is in Australia's mesic zone. So you won't find orchids in the driest areas, and they are most diverse in eastern and southwestern Australia. So here are just a couple of slides to show the range of morphologies. They can be really vastly different in their flower shape and form, and that has puzzled biologists and botanists a long time, and it was really difficult to understand how those different orchids, which in themselves form very distinct groups. For example, spider orchids is a hugely diverse group. They look very similar in itself, but not so similar than to other Australian orchids, how they would be related to each other. And then we have some really peculiar ones like the flying duck orchids and the hammer orchids, the underground orchid, which is a mycoheterotrophic species that relies solely on nutrients from the fungal partner. So in our research, we are interested in understanding how the Australian orchid flora was assembled over time and evolved over time, with questions, where did Australia's orchids come from, over which evolutionary time span did they evolve, and what are the evolutionary relationships among Australian orchids. So for that, we have assembled large genomic data sets, and these are based on collections data. Our initial aim was to sequence 1,000 Australian orchids, and we have now sequenced over 2,200. And in that project, we carried out genome skimming, which is a technique where the highly abundant DNA fraction is sequenced, and that is mainly the chloroplast for plants. So the first study that I want to present is on the evolutionary relationships and range evolution of the greenhood orchids. Greenhood orchids are the second largest subtribe in the Australian orchid flora, with over 300 species. And they sit in that orchidoidy branch in the tree. The distribution is very typical for Australian orchids, again, along that rainfall gradient in the mesic zone. They also occur in New Zealand and New Guinea and Caledonia. So extend into the Australasian region. While they are not the most beautiful orchids to humans, they have great diversity of shapes and forms, and in particular in pollination syndromes, as you see on those slides, some of those species have the outer part downwards facing, and others upwards. And the ones that are upwards are trap flowers, and the other ones also attract insects, but more by resembling a female insect. As you can see here, maybe it has little features. So we carried out a phylogenetic study using those plastid markers, which revealed the evolutionary relationships in this largely diverse group. And we found that 11 major evolutionary lineages that coincided with major groups that were taxonomically also recognized at sectional level, so within a genus, but by other workers also at a generic level. So we won't focus too much on taxonomic names here. We'll look at the evolution through time. This is a simplified graph of a time tree, just focusing on the different epochs and when the greenwood orchids evolved. So the origin of the greenwood orchids is at the boundary between the Eocene and Oligocene, which is a time when the Australian continent just was in the process of drifting from Antarctica, to which it had detached, and drifting northwards. That is the evolutionary origin, and the closest relatives of the greenwood orchids are actually in South America and were connected through Antarctica, which at that stage, or before it became all frozen, was still vegetated. So this was a mechanism by which the greenwood orchids were isolated from their relatives. Then in the Miocene, we find that all 12 evolutionary lineages have emerged, and it's a phase in the Australian history of the continent in which is characterized by increased aridification. These are small terrestrials that benefit from open landscapes versus forested landscapes. And so here they obtained more opportunity to diversify during this period of time. Then in terms of species richness, it is very apparent that those little triangles only emerged very late in the Pliocene and Pleistocene, so from 5 million years onwards. So our current diversity is relatively recent of those different evolutionary groups. And that is often interpreted as a consequence of frequent climatic oscillation that we saw during those times, which led to range expansions and range constrictions that led to increased isolation and secondary contacts. So in terms of where the greenwood orchids came from, we dated the phylogeny and did an ancestral range reconstruction, and these are the results from that reconstruction. So the origin of the greenwood orchids was reconstructed as Australian. So the ancestor embarked on Australia about 33 million years ago in the Oligocene. And then the diversification took place mainly on the Australian continent. We were also able to trace floral exchange and expansion range shifts to other areas in Australia. So, for example, from Eastern Australia, which is today's center of diversity, to Southwest Australia, which started only in the late Miocene, about 10 million years ago. So with that, I would like to come to the last study for the evolutionary part. and looking at the tribe Diuridae. So the tribe Diuridae is one of the largest, or is the largest Australian lineage, and comprises around 60% of the Australian orchid diversity. Together with the greenwood orchids, they make up 80% of our orchid diversity, and then the remaining about 20% are the epiphytic orchids. They are also geophytic herbs, so they also stay or hibernate underground with their tubers. 9 subtribes are distinguished, and around 979 species. As you can see from the distribution map, they are have an Australasian center of diversity, but expand quite significantly into neighboring regions, New Zealand, New Caledonia, New Guinea, which would be Australasian, but also into the Malaysian and Asian region, and also into India. This distribution pattern has always puzzled orchid researchers, and has left us wondering where those orchids came from, whether they originated in Asia or Australasia. And there are some really awkward Diuridae species on New Caledonia. So there was also the question whether they might have originated in New Caledonia, as New Caledonia has some other really relict plants as well. So we investigated the evolutionary relationships of Diuridae, again with a plastid data set through genome skimming, here with 243 samples representing all genera that are recognized, and found that we had a really well resolved tree with 2 little weaknesses at the base of the tree here, where the evolutionary relationships between this group, Cryptostylidinae, and the donkey orchids, Diuridae, was not quite certain. And this particular log branch, which is for the species in the underground orchids, the Rhizonthelinae, which have suffered a lot of plastome degradation because they don't need to use their plastid genes anymore. The one species where the plastid genome was fully sequenced showed that only 23 genes remained out of normally over 100. So therefore, its position was also not quite well supported, but we are quite confident that it is a relative of this group Rhizonthelinae, which also has some microheterotrophic species. So we collaborated with the GAP consortium, to which we contributed the orchid data, collaborated with PAFTOL to assemble the orchid tree of life globally, and so contributed the Australasian data set to this large endeavor. And this is a paper that is currently in preprint and has also been included as a chapter in the Q report that I presented at the start of this talk. So looking at the origin of orchids on a global scale, the study found that the ancestor of the extant orchids, which is was about lived about 83 million years ago in the Cretaceous, so at the time that the dinosaurs were still around, and that the ancestor of the extant Diuridae, our largest group of orchids, emerged around 42 million years ago, so in the Eocene, at a similar time as the greenwood orchids, actually. So they underwent a very similar evolutionary history in long isolation with on the Australian continent, diversifying to the different branches, lineages, also mainly in the Miocene, with the opening up of opportunities through the opening of vegetation. An interesting result of the study was about diversification dynamics in orchids, so speciation rates. And the study first identified highly diverse regions in the world where orchid diversity is concentrated, which was in Central America, the northern part of South America, then of course Asia, New Guinea, and Western and Southeastern Australia. Then they studied how quickly those different groups evolved. And it was really peculiar, or it is peculiar, that while most of the species-rich regions of the world also have elevated diversification rates, this is not the case so much for the Australian orchids, which is interpreted as being associated with the more geological stability of the Australian continent, whereas the other regions are affected by mountain rises like New Guinea and the Andes and Central America, which made the diversification rates speed up. Australia doesn't have this pattern, and that shows that species probably accumulated over a longer time span to the diversity that we have today. So in summary, for those evolutionary studies, we now see that Australian terrestrial orchids have largely diversified in isolation on the Australian continent while it was drifting to its current position, and that they have had long evolutionary time spans to adapt to the Australian environment, and that they are an important source of neighboring orchid floras. I want to switch gears and present on one study that we've done, and it's an exemplary study for our work on threatened species. So we have a lot of Australian orchid species that are very recently evolved and very difficult to tell apart and to draw boundaries around taxonomically to really understand what constitutes a species. When new species are described and the taxonomist is of the opinion that something is a new species, and they have small distribution, that flags them as being, probably endangered, and then conservation assessment is made. And in this case, there were two widespread species, Corybus barbary and Corybus acotiniflorus. And then a new species was described with a very small distribution. And it was in an area where there was development of a major road. So it was immediately listed as vulnerable, and also was listed to have some more work around understanding its genetic diversity and its species delimitation. Now, when you look at this representation of the three species, as a lay person or botanically inclined person, you would say, yeah, I can see one is white, the other one is red, and the other one is more greenish. When you look at the broader diversity, you would have probably difficulties, and I do as well, to draw a line or group those species into, or these plants into their correct species, because it seems that there is a bit of a gradient and it's not so clear cut as the descriptions of the species make believe or convey. So that was the reason why we undertook a genetic study on this group. here are the distributions of the three species. So Corymbus acotoniflorus has actually a very large distribution and occurs from here, from around Cairns, until Tasmania, whereas Corymbus arbore co-occurs for long stretches with Corymbus acotoniflorus, but doesn't go as far south. And the dark dot here is where they all three co-occur, Dowlingii, in a really restricted area, together with acotoniflorus and barberry. So they are sympatric. Our study, and that is based on genotyping by sequencing dataset with technology called ddRAD, found that the branches that we have on the evolutionary tree are not consistent with the classification that would distinguish three different species. As you can see, we don't have three clades that would have the different species, but there's more of a mix, and also even the other species are also not forming a consistent evolutionary lineage. So that was an indication that probably the taxonomic concepts are not quite right. We did a network analysis. This is another representation of the data set that shows conflicts in the data sets, and found that there is conflict that might show hybridization between those three species. And further analysis confirmed that there is gene flow, but not only between one and the other species, but between all three species, which is a very complicated system. So the next step is that we have obtained funding to do a more large-scale study across the whole distribution of the two more widely distributed species to understand better what is going on in this group. So with this, I would like to acknowledge the orchid research team at the Australian Tropical Herbarium with currently Lalita Simpson and Lars Nauheimer as postdocs. Stephanie Gödetz has just done a very beautiful master study on a microheterotrophic orchid genus, Consolata nanjala, sitting here with us, PhD student who will work on diversification, dynamics, and trait evolution in Australian orchids, and Sophie Gister, who is helping us with lab work. And then previous postdocs and students that have contributed to work that I presented today. And with that, I would like to acknowledge our collaborators, all the technical assistants from various collaborators, and our funding partners that enable us to do that research. And I would like to thank you for your attention.